back again for what I'm hoping is the second to last, the penultimate lecture on the immune system. So last time, you and we were talking about T cells, T lymphocytes. So we got to move on and talk about B cells, B lymphocytes. B lymphocytes, of course, are born in the red bone marrow. There's some red bone marrow right there. Nice. Now, B lymphocytes are born there. Same place as T lymphocytes. However, remember the T lymphocytes migrate to the thymus for their maturation, for their finishing school. B lymphocytes are going to mature in the bone marrow. So they're going to acquire their surface receptors. They're going to become immunocompetent. We're going to make sure they're self-tolerant. They don't attack our own tissues. That all happens in the bone marrow, the red bone marrow for B lymphocytes. Okay. So once we got lymphocytes made, we're going to make about 1 million different varieties. Each different variety, there'll be a group that have a receptor in common. There'll be 1 million different groups. Let us respond to perhaps 1 million different antigens. Now, once we have these immunocompetent lymphocytes, Bs and Ts, they're not going to stay in their schools, their respective schools, the bone marrow and the thymus. They're going to leave. They're going to go to lymph nodes. See all these nice lymph nodes here, colorful. To the spleen, and to the palatine tonsils, as well as other tonsils, as well as to your tracts, because those are the places where antigen challenge is going to occur. Those are the places where lymphocytes are going to meet up with antigens and are going to try and deal with them. All right, so as we talk about this adaptive immunity, this immunity that can get better and better and better with each exposure, we got to realize there are 2 branches to it: antibody mediated and cell mediated. Let's rock through those. Antibody mediated immunity. It is how your body's going to deal with extracellular pathogens. By extracellular, I mean outside of cells. You knew that's what I meant. And basically what I mean by that, if you're outside of cells, you're just swimming in body fluids. Body fluids like tissue fluid, body fluids like plasma, lymph, pee, etc. And an old-fashioned word for a body fluid is a humor. So an old-fashioned term for antibody-mediated immunity is humoral immunity. Now, of course, antibodies are made by plasma cells, which are derivatives of B lymphocytes. All right, now, once a pathogen has invaded a cell, well, antibodies are useless. So intracellular pathogens, they might think they're protected, right? But they're not, because we have killer T cells, aka cytotoxic T cells. And these guys are going to be responsible for cell-mediated immunity, which is going to deal with intracellular pathogens, and also, by the way, help us kill cancer cells too. Okay, both of those branches, in fact, the entire adaptive immunity, is going to be under the regulation of helper T cells. Of course, you know where helper T cells are born, you know where they mature, you know what they have in their surface. Answer these 3 questions on your own. Let's keep going. All right, check this out. Here is our naive helper T cell. There he is, okay? He's out of the thymus. He's immunocompetent. He's got the receptors, many copies of one specific receptor. He is being shown an antigen. Here's the antigen. Here, 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 and here. All right? The antigen is being displayed by an APC, an antigen presenting cell. It is doing the presenting by means of its MHC2 protein. Remember, an antigen presenting cell is most likely a macrophage. So a macrophage engulfed something foreign, chopped it up, held up the piece on its MHC2. This macrophage probably went to a lymph node because it needed to run into the right T cell, the T cell with the right receptor. And in response to this interaction, this single solitary T cell here is going to undergo mitosis. And in mitosis, we're going to get active helper T cells. Let's say these are active helper T cells. They're going to deal with the the here and now infection. And this guy over here, we're calling him a memory T cell. He's going to hang out and be ready for next time. Okay, so what we're doing here is we're starting our adaptive immune process by turning on helper T cells. And this makes sense because helper T cells are in charge of all the other aspects. Now, once a helper T cell is activated, the helper T cell is going to make a lot of chemicals called cytokines. Cytokines are going to regulate and activate cells of both the adaptive and innate immune system. So we're talking about killer T cells, we're talking about B lymphocytes, we're talking about plasma cells, we're talking about macrophages, dendritic cells, NK cells, eosinophils, basophils, neutrophils, monocytes, etc. A lot of the cytokines, notice there's an example, it says IL-2. The IL, a lot of cytokines have this IL abbreviation. IL stands for interleukin. That word makes sense because inter, between, leukin, white blood cells. So, cytokines released by activated helper T cells are going to stimulate the activity, the multiplication of other immune cells to let us deal with whatever nasty infection or not so nasty infection, whatever foreign thing is in us. All right, now meanwhile, while all that is occurring, okay, the macrophage presented the antigen to the helper T cell underwent mitosis, made active helper T cells, memory helper T cells. Can you guys see my hands demonstrate all this? Probably not. I don't know why I'm doing that. Meanwhile, at the same time, that pathogen, that antigen, is floating around in your body fluids. I mean, a macrophage found it. There's going to be more. In this case, we have a B lymphocyte right here, a naive, immunocompetent B lymphocyte. He has receptors on him. These receptors are totally specific for that same antigen. Okay? Once the B lymphocyte binds the antigen, he'll swallow it, he'll chop it up, he'll display it on his surface. Okay? He'll display it on his surface in order to present it to our helper T cell here. What the B lymphocyte is doing is it is basically saying to the helper T cell, and of course it's got to be the right helper T cell, which we just made a bunch of these activated ones. What the B cell is saying is, hey, listen, I just killed this thing. I just killed this guy right here. I want to make an army and kill everything that looks like it. Do I have your permission? Okay, because this is a dangerous thing. Cells that kill other cells. This needs to be regulated. And by having the B cell check with the helper T cell, it gives us a level of safety and control. So, the B lymphocyte is showing the activated helper T cell the antigen it engulfed by means of its MHC2 protein. And the activated helper T cell has got to have the right receptor. And in this case, the activated helper T cell is saying, yes, go ahead. And he's saying that by releasing cytokines. Notice IL again, interleukin, between white blood cells. Now, the activated B lymphocyte is going to undergo mitosis. It's going to proliferate, making plasma cells, making memory B cells. Check this out. We made 3 plasma cells, 2 memory cells. The memory cells are for later on. The plasma cells are for right now. Plasma cells make antibodies. Later on, if these memory cells bind the same antigen, we'll make more memory cells, more plasma cells, which will make even more antibodies, and it'll be more awesome the second time around. All right, I think that's it for this time. I think so. See you later.